Okay, good morning, students. Am I audible now?
Yes, ma'am, you're audible. Good morning.
There was something wrong with my earphones, so I guess they were not working right. Okay, so I guess almost 14 students have joined. So few are still to join. So this was your first class?
Yes, ma'am.
Yes, ma'am.
Okay, they might be joining, right? So yesterday we discussed about the course postulate, and we thought that today we'll have a discussion session on this course postulate. So if as a opening remarks, I'll just say that in the beginning of the 19th century, the Koch's postulates were very important. They were discovered or they were given by Robert Koch, a bacteriologist, and he has enormous work in bacteria, a lot of diseases, and some additions in the microscopy are also being given by him, right? So at that time, what was the basic reason behind this postulate was that he wanted to say that how we know that a particular pathogen is causing a particular disease, right? So for that we need to follow some steps to prove that basically the organism, one particular organism is responsible for causing disease in plants, humans, or animals, right? Bacteria or a fungi, right? So he laid down some postulates, and the fulfillment of those postulates was important to prove the pathogenicity of the microorganisms or pathogens at that time. Though when he was himself working, he realized that these postulates were not being there were certain exceptions to these postulates, and it was not important that all the pathogens they require to follow these postulates, right? So he himself identified that's, for example, certain asymptomatic diseases such as cholera was sometimes called by certain asymptomatic bacteria or other causes, so in or typhoid also, right? So in those asymptomatic infection, we know the symptoms, right? We don't know what kind of a symptoms because they are not causing any symptoms. So how do we identify, or how do we prove the similar symptoms caused by the pathogen if we are isolating them, right? So he at that time also it was quite known that in all the cases this cannot be followed. Similarly, other viral infections such as polio or HIV or hepatitis C, in these cases also sometimes symptoms are not very prominently shown. So these are also exception to the Koch postulate. And then it is the second postulates which states that we need to grow a pathogen in artificial media. So this postulate definitely doesn't go for the prions or for the viruses pathogen, because viruses we cannot grow in artificial media. Yes, we need to maintain them on a live host, then we can isolate them, then we can go and sequence them, and we can see that whether it's the same pathogen, but definitely we cannot grow them in a pure culture, right? So there were certain points that due to which these postulates, but still despite these kind of a drawbacks or these kind of shortcomings, these postulates are still being or were still being used to identify the pathogenicity of a particular pathogen. So when I did my MSc, I was working on a septoria leaf spot of a chrysanthemum, which is a fungal pathogen. So at that time, I followed these postulates and I proved the pathogenicity of this postulate and proved that this is the pathogen which is causing this disease. So I did myself these experiments, and it took me almost around 2 months to prove these things. So at that time also, we were using Koch postulates to identify a particular pathogen or to be known as the causal agent of a particular pathogen, right? So yes, we cannot say that these we need to follow these postulates to prove the to identify the pathogen in all the cases, but still today these are being used to identify. And now science has, or technology has improved a lot. Earlier time we were not having molecular technique. Earlier time the sequencing was a dream, especially for human pathogen. Also it started very late, right? Sequencing or a molecular technique such as a RT-PCR or a PCR. So they were not having any options. Right now I might not need to go for a post postulate. I'll just isolate a fungus. I'll not even isolate a fungus, or I'll not even isolate a virus. I'll just go and isolate DNA and RNA. I'll use certain primers for which I have a doubt. I'll use PCR for the amplification. I'll clone it. I'll sequence it. I'll see what is this, whether it's a similar strain, whether there are certain changes in the this particular pathogen that I'm working on, or this is how different strains of a coronavirus are being identified. This is how we know where are the mutations in the different in the genome of the coronavirus, right? So we are not relying on these techniques, but still, certain sometimes a biological experiments such as Coast postulates, it also come under the biological experiments. So certain biological experiments, they were very important to study the, to study a particular pathogen, to know about its incubation period, because if we'll artificially inoculate, we'll know the first, when will the first symptom will appear. So we'll be able to tell, okay, this is the incubation period for this particular pathogen. So those techniques were still important. And now also these techniques are being used. Yes, because not everywhere we have molecular technique, not everywhere the sequencing is a easy code. But yes, in higher end labs, definitely we more rely on the molecular methods or on the sequencing, because if we have funds, we can easily go for these techniques, and we are very sure after sequencing, when we know the genome of a particular pathogen, so we'll be able to tell about the strain. Also we'll be able to tell about the variations, the combinations, mutations, whatever they are present in a particular pathogen. So definitely they are the best, but still biological experiments are being considered because they are the basics, right? This is how we studied them. In the earlier time, viruses were also not, they were not being amplified by using PCR. There were certain biological assays or local lesion assays, or the kind of the symptoms they produce after artificial inoculation. So there were a number of biological assays to identify the viral pathogens also, when the molecular techniques were not readily available, right? So now over to you all. So we can just start from the top or from the bottom, or just whatever, in what way the names are appearing in the team. So I'll just start. So you just tell, what have you read about Coach postulates, since today was a discussion class. So just whatever you have read about a particular this topic, right? Okay, so Adhish Mishra. Adhish, can you please switch on your camera and speak? Okay, Adhish is not responding. Ayushi, can we move on to you?
Ma'am, am I visible?
Yeah, Ayush, you are visible. I can see you. Yes, go on.
Yes, ma'am. So, as we all know, Robert Koch was a German physician and bacteriologist. So, he identified causative agents of anthrax, cholera, and tuberculosis. So, he applied the postulates to establish the etiology of anthrax and tuberculosis, but they have been generalized to other diseases. Um, the four criteria designed by Koch and Friedrich Loeffler in 1884 to establish a cause relationship between a causative microbe and a disease, they are known as Koch's postulates. So the four criteria that were established by Koch to identify the causative agent of a particular disease includes that the microorganism or the other pathogen, it must be present in all cases of the disease. The pathogen can be isolated from the diseased host and grown in pure culture. the pathogen from the pure culture must cause the disease when inoculated into a healthy or susceptible animal or plant, or we can say susceptible host. The pathogen, it must be re-isolated from the new host and show the same or original symptoms the same originally inoculated pathogens. So, some few limitations for Koch's postulates are that the causative organism might not be present in every stage of the disease. Like, for example, Vibrio cholera, the infected individuals in infected individuals, it might not be present at the time of extreme vomiting or diarrhea, because it releases toxins, which causes the diarrhea even in the absence of the organism. Again, many viruses, they do not cause illness in all infected individuals, which is the requirement of postulate 1. For example, if we talk about poliovirus, it which causes paralytic disease in about 1% of those who are infected. And as uh, as we all know that particular bacteria like leprosy and many other organisms like viruses or uh, they cannot be grown in pure culture in the laboratory. And yeah, that's it, ma'am.
Okay, very good, Ayushi. You mentioned few points which were very important, such as Vibrio cholera, that it is not important that pathogens should be present in the host all the time, and the examples that you gave about the other pathogens such as viruses and other, where we cannot culture them in artificial media, right? So we cannot culture even the Mycobacterium leprae, bacteria that cause leprosy. We cannot culture that bacteria in artificial media. So in those cases, the coach postulates they somewhere they have a shortcomings, or we say they are failing, right? Thank you, Ayushi. Very good. Yes, can we move on to next, Sam?
Yes, ma'am, am I audible?
Yeah, Sam, you're audible.
Uh, okay, ma'am, like Ayushi explained most of the exceptions. So
Yeah.
I was looking through postulates and its exceptions. So I found some more exceptions and
Mm-hmm.
limitation. So
Yeah.
like one was that there are very limited animal models, as not all humans can be tested for this, because they could some microorganisms are harmful and it would be unethical to test on humans.
Mm-hmm.
Then oncogenic bacteria and viruses like papillomavirus, these can cause cancer, but in some individuals and not in others. So you can't call, so the disease caused by them is not, like, you can't say the Cox postulate won't be applicable here. Then prion diseases like mad cow diseases and all. And then there are also some genetic resistance for those people who suffer from sickle cell anemia. They won't be infected by malaria.
Mm-hmm.
So at that point, the third postulate would not be applicable here. And I think the last exception that I found was of a protozoan called Toxoplasma gondii. And research showed that this protozoan can cause psychological disorders like related with anxiety and autism, schizophrenia, and all. So again, psychological disorder depends on the person and how the rate of how they are responding to that psychological disease. So Cox postulate won't be applicable here also, because different individuals react to stress differently.
Mm-hmm. Okay, okay, Sam. They were good point that you mentioned that other than that, other diseases or about other pathogens that Ayushi mentioned, that there are other exceptions also where coach postulates cannot be considered to be fully applicable. Yes, right. Can we have another one? Ishita Arora.
Good morning, ma'am.
Good morning, Ishita.
Ma'am,
Yeah, you're audible. You can just start. Okay.
okay,
You are
ma'am. Ma'am,
vis-
so according to Koch post, yes, ma'am. Ma'am, Koch postulate, basically, it is one of the postulates according to which we can study about the diseases, how diseases are caused in organisms. The organism is infected due to some disease. Ma'am, in this postulate, microorganisms which organisms suffering from disease are due to microbes, or we can also say due to the pathogens. Ma'am, in this ma'am, microorganisms must be observed in every case where any of the organism is suffering from disease. And ma'am, here there are many examples, as Ayushi already said. Symptoms and the disease can be easily identified by the symptoms and the signs that one is showing. Ma'am, according to this postulate, there are many different, according to this postulate, we can also prove that that particular disease is caused by which organism.
Okay, Ishita. And first thing, इतनी बारी मैडम बोलने की जरूरत नहीं है. You're telling not only to me, you're telling to the whole class. So it is fine that you don't have
Sorry,
to say
ma'am, actually.
ma'am so many
Sorry.
times, right? So it's just fine. Okay, Shita. That's good, right? Can we move on to next? We have Janvi Singh here, right? Yes, Janvi. Are you able to unmute yourself? No. Yes, Kshitij, can we move on to you then? No. Manaswani, then you, I guess.
Yes, ma'am. Am I audible?
Yeah, you're audible, man.
Ma'am, there's some issue with my camera, so I'm not
Okay,
able to
fine.
switch it on.
It
Okay,
is fine.
so this is mostly from whatever I read and from what we had in class yesterday. But I wanted to talk a little bit about the importance of the postulates and why was it a requirement? At the end of the day, it was just four statements, but for some reason, it had such a big significance. So
Yes.
from what I read, it was because it was one of the first ways to properly establish a relationship between a particular pathogen and the disease that it caused. And I also feel that it also helped kind of explain the pathogen triangle, that triangle of infection, because the pathogen won't be exactly the same, or the disease won't be exactly the same in every host. So when we reproduce it in the next host, let's say the symptoms are a little different, but we're still able to isolate the pathogen from another host, then it could mean that the disease can also manifest in different ways based on the tolerance or susceptibility of the host as well. And discoveries like this definitely help study the pathogen better, and that is very important because only if we properly understand the disease and the causative organism will we be able to come up with proper treatment methods. So this is mostly what I read.
Yes, Manaswini, you're right actually. So, in order to yes, different hosts they definitely react differently to the pathogen. So this is a very important point here. So, see in case of the plants, sometime we cannot go and produce the same disease on the similar kind of a plant, especially for example, yesterday we discussed an example of a crown rot of an apple which is being caused by a fungus. So that was easy because we could take an apple and we could directly infect the apple, and the symptoms are being produced. But sometimes the symptoms are not seen on the fruit, but they are seen on the branches, or they are seen on the barks, or the leaves, right? So in those cases we cannot just go and take a grown up tree and we inoculate that. No, in those cases we need some host plant, or some host plant where the similar virus go I talk virus more, because but say where the similar pathogen can go and infect. So there changes or variation in the symptoms can be seen, because on different kind of a host plant the pathogen has a different impact, right? So yes, this is an important point here, that we need to see this, we need to take into consideration this point also. But the time when we are talking about the early 19th century, right? The time when these postulates were discovered, there was no other mean to identify a particular pathogen, or the means were very less. So in that time, definitely these postulates, they, with exceptions, yes, exceptions are there, always there, but they definitely played a very important role to identify a particular diseases or a particular pathogens, or to see whether the disease is being caused by a same pathogen, or we are we able to reproduce the disease on the host plant, are we able to grow them in a pure culture where we are talking about the fungal pathogen. Yes, so this is important here. Yes, Namrita, can we move on to you?
Yes, ma'am. So exceptions of Coates' postulates were that some microorganisms that cause disease have never been cultivated under laboratory conditions. Second, some diseases are caused by several microorganisms. Third, some microorganisms cause several diseases. And fourth, some diseases have variable signs and symptoms between patients. And fifth was ethical consideration prohibit experiments when humans are the only host for a particular disease.
Okay, okay, Namrita, thank you so much. Can we move on to next student? We have Nandini. Namrita के बाद है Nandini. Yes, Nandini.
Good morning, ma'am.
Good morning.
Ma'am, I wanted to talk about why the pure cultures are important cost postulate. Because mainly the only exception is because of the
Yeah.
pure cultures. So,
Yeah.
cost research and methods help link the casual nature of microbes to certain diseases, such as anthrax. So, ma'am, when later these postulates were developed, the pure cultures were mainly used to allow the pure isolation of a microbe, which was very important in understanding how each and every microbe reacts and how it contributes to a disease. So, I mean, it's already widely accepted that there are like some species that do not cause disease, and they do not fulfill the Koch postulates, since Mycobacterium leprae and Treponema pallidum, which are implicated in leprosy and syphilis, they are not grown in culture medium. So that's it, ma'am.
Okay, Nandini, yeah, that's right that we are not able to grow all the pathogens in a pure culture, but then pure culture has its own significance. So whenever we are talking about a pure culture, that means we are having a pure culture of a particular pathogen. So that culture, it was important to even to know about the pathogen, that how pathogen is looking, what kind of a spore it is producing. If it is a fungal pathogen, how does it look under the microscope or electron microscope or compound microscope. So to study the various characteristics of the pathogen, also the pure culture was important. And sometimes when we are doing certain kind of a host pathogen studies, in those cases also we require this kind of a pure culture to see the what kind of a symptoms are being produced, or how different hosts, they are resistant or they are tolerant or they are susceptible to particular pathogens. So it definitely plays a important role, pure culture. Yes. Yes,
Yes,
after Nandini, can we have a Nehal? Yes, Nehal.
Good morning, ma'am.
Good morning, Nehal.
Ma'am, as you said that technology has become so advanced, and
Hmm.
there are different methods for the identification and detection of microbes that are causing disease in a particular plant. So one of them is nucleic acid-based microbial detection method. So this method enable the identification of microbes that are associated with a particular disease. These methods are very sensitive and can detect very low level of viruses in a healthy organisms. So this technique basically involves culturing and measuring the growth of individual microbes using they might either use non-selective media such as trypticase, soy agar, or they can use selective media to detect a particular bacterial species. And at lower detection level, this method require a lengthy pre-enriched step to increase the number of target bacteria before the detection. And this method have an advantage, that is they are they have high specificity, high sensitivity, and they have the ability to quantify the pathogen biomass.
Okay, Nehal. So yes, you talked about the various additions or various changes in the different medias which can be done in order to culture the bacteria or in order to increase the chances of culturing of a particular bacteria. Yes, it is important. And see, since Coase postulates were somewhere we started using them in the 19th century, and after that, lot of changes and lot of additions according to the pathogens or according to the different Coase and pathogens, they are being incorporated to study these postulates basically. So yes, you will find if you'll go through different papers, so in various papers you'll find there are certain amendments which are being made according to the requirement of a particular study. So yes, it is not exactly, they are not exactly followed as they are written. There are a lot of changes or there are a lot of amendments being done in order to reproduce the result. Okay, so after Nehal we have a Sahiba.
Good morning, ma'am.
Good morning.
Ma'am, I would like to summarize everything from the
Okay,
starting.
fine, no problem.
So, cautious postulates are four criteria that were designed to establish a causative relationship between a microbe and a disease. The postulates were formulated by Robert Koch in 1884. So the four postulates were that the organisms must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. The second one was the microorganism must be isolated from a diseased organism and grown in pure culture. Third one was that the cultured microorganism should cause disease when introduced into a healthy organism. And the fourth one was that the microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. So there were many exceptions for these postulates. So one of the exception was that some microorganisms that cause disease have never been cultivated under laboratory conditions. And incautious postulates the blood-borne pathogenic microorganisms are cultured in artificial media. So, however, some microorganisms cannot multiply in artificial growth media. For example, lepre can only grow in armadillos, a type. It is a type of New World placental mammal. That's it, ma'am.
Okay, okay, Sahiba. Can we move on to next? Next we have Sanjana. Yes, Sanjana.
Good morning, ma'am. So, Robert Koch gave four postulates, which were published in 1890, which were a set of criteria that established whether a particular organism is the cause of a particular disease or not. So, some of the exceptions to Koch's postulates were
Sanjana,
first.
can you just change the location of your mic or something because I cannot hear you clearly. It's vibrating. Your voice is breaking.
Is it fine now?
Yeah, yeah, it seems fine.
Okay. So, Robert Koch gave four postulates that were published in 1890, which were a set of criteria that established whether a particular organism is the cause of a particular disease or not. And some exceptions to Koch's postulates were first, as Sahiba mentioned, that some organisms that cause disease have never been cultivated under laboratory conditions. For example, lepre, which can only grow in armadillos, that is a type of New World placental mammal. Second was that some diseases are caused by several microorganisms. So, therefore, microorganisms cause distinct signs and symptoms in the host, for example, as in tetanus and diphtheria. However, several microorganisms may elicit the same disease conditions in various situations. For example, kidney inflammation, that is in nephritis, and pneumonia can be caused by several types of pathogens. Another exception was that some microorganisms cause several diseases. For example, tuberculosis can cause several diseases, conditions in the lungs, internal organs, bones, skin, etc. And also, S. pyogenes can cause sore throat, scarlet fever, and osteomyelitis, the symptoms of M. tuberculosis. Also, some diseases have variable signs and symptoms among patients. For example, tetanus shows variable disease conditions among different individuals. And also, ethical, for example, in HIV, ethical considerations also prohibit the experiments for when we are dealing with humans. When humans are the only host for a particular disease, if there is an investigation or research on HIV that infects humans, it is unethical to purposely infect a human with HIV. And also, ma'am, I read about the second postulate of Koch that states that the bacteria must be isolated from the host with the disease and grown in pure culture. So the article gave three instances. First, it was that suppose an organism A could still be present and isolable when the individual developed a subsequent disease due to organism B. That means the pathogen that we are isolating is not the actual one. Second circumstance was that where an organism A resulted in a completely asymptomatic subclinical infection, and at the same time the patient was infected with organism B, which was the real culprit in producing the clinical manifestation. The third circumstance was that where organism A exists in a completely latent state until organism B infects the individual, producing an illness, but also reactivating organism A from latency to active asymptomatic multiplication. So many an example of this is that many bacterial forms, especially intracellular organisms such as tubercle bacillus, brucella, parasitic infections like malaria, and many viruses, especially of the herpes family, are examples of these three possible circumstances.
Okay, Sanjana, that was a very good detailed account you've given, and with other examples also, right? So can we have Shakti now? Shakti?
Yes, ma'am. Am I audible? Hello.
Yes, Shakti, you're audible.
Okay, so I read about modification of Koch postulates. So these were done by Fredericks and Rellman in 1998, and these were done by nucleic acid-based pathogens, like which have DNA or RNA. So it said that a nucleic acid sequence of known pathogen should be found associated in most of the cases with disease, and it can be found in those organisms which are having symptoms or which are affected by the disease. And it also says that pathogen associated nucleic acid sequences may or may not be present in fewer number from the host or tissue without disease. Like when we compare two tissues, one which is affected with the disease, then the nucleic acid which is associated with disease will have in higher number as compared to the other tissue which is not affected. And also, when the copy number of the pathogen associated nucleic acid sequences should decrease or become undetectable in case of resolution of disease. That means if suppose the inflammation is decreasing, so then the copy number of the pathogen associated nucleic acid sequence should be decreased, or it will become undetectable. Yeah, so that's it.
Okay, Shakti. So I was just wondering before this also that nobody mentioned about the Fredericks and Rellman, because he was the person who modified post postulates for the 21st century, and he gave a lot of he included a lot of new things and the lot of new techniques basically that where we can work with the genome and we can see the changes which are being caused in a particular genome of a particular pathogen. Yes. Okay, Shakti, good. So can we have Shivani now? Shivani.
Good morning, ma'am.
Devanshi, I'll come to you. I guess I forgot your name or you was not there in the queue at that time, I guess. So I'll come to you
Ma'am,
later.
actually, I just joined because
Yeah,
I
that's
wasn't able to join.
okay, fine. That's why I didn't took your name, right? Okay, I'll come to you later, right? Yeah.
Okay, ma'am.
Yes, Shivani, go on.
Good morning, ma'am. Actually, my points are already been discussed. Like, it's all covered. Hello, ma'am. Am I audible?
Yes, you're audible, Shivani. You're audible.
Hello.
Your voice is breaking though, Shivani, I guess, but you are audible. Am I audible or not? The network is showing bad at my end also. Hello? I guess it's
Ma'am,
gone.
yes, you are audible.
Ma'am, you're
Okay,
audible.
fine. Yes, Shivani, now you can go for.
Yes, ma'am. Good morning, ma'am.
Good morning.
Ma'am, actually, the points that I was going to like, it's all been summarized. Like everyone said, all of that.
It's okay, fine, but still I would like to hear something from you.
Yes, ma'am.
सबने तो क्वेश्चन आंसर लिखेंगे ना पेपर में, लेकिन तुम अपना
Yes,
आंसर तो तुम
ma'am.
लिखोगे,
Yes,
उसी के मार्क्स मिलेंगे. Yes.
ma'am. So basically, the coach postulates were the like basic criterias of how do we know that a particular or a given pathogen cause a specific diseases. So there were four coach postulates. The first one was the pathogen must be present in every case of the disease. I'm sorry for the voice. So first one was the pathogen must be present in every case of the disease, which was later on proved like it came to an exception after coach himself discovered the cholera carrier. The second one was The second one was the pathogen must be isolated from the disease host and grown in a pure culture, which was again everyone discussed right now that pure culture, like viruses cannot be grown in pure culture. The third one was the specific disease must be reproduced when a pure culture of pathogen is inoculated into a healthy susceptible host. This can there can be exception like there must they can be non-infectious but carriers as well. The fourth one was the pathogen must be recoverable from the experimentally infected host. Ma'am, that's
Okay, Shivani. Okay, fine. We have Shivani के बाद है Tanvi. Yes, Tanvi.
So, I want to tell about the scientific contributions of the Koch. So, first of all, Robert Koch made two important developments in the microscopy. So, he was the first to use an oil immersion lens and a condenser. That from that we can see the small objects, and he also was the first to effectively use the microphotography for microscopic observation, and he used the bacterial staining using methylene blue dye, and he in an attempt to grow bacteria, Koch began to use solid nutrients like potato slices, and he was also the one who developed the petri dish, and he also contributed in the anthrax. So he worked on anthrax, discovering the causative agent of the fatal disease to be Bacillus anthracis, and he published the discovery in his book that was the etiology of anthrax disease based on the developmental history of Bacillus anthracis. And there was also one discovery on tuberculosis that he did while he was working. So during his time, Robert Koch became interested in the tuberculosis research. So he contributed in the tuberculosis also, and in cholera also he contributed. And lastly, tuberculosis treatment and tuberculin. Koch gave much of his research attention on tuberculosis throughout his career, and there was that Koch observed the phenomenon of acquired immunity. So while he was doing his experiment, who had been brought to New Guinea fell sick immediately the longer they had stayed in the country. So he saw in his experiment that there was some acquired immunity while he was doing the experiment on guinea pig. And there was four postulates of Koch that the organism must always be present in every case of disease, but not in the healthy individuals. And second, that the organism must be isolated from a diseased individual and grown in pure culture, and the pure culture must cause the same disease when inoculated into a healthy or susceptible individuals. And fourth, the same pathogen must be isolated from the experimentally infected individuals. That's it, ma'am.
Okay, so yes, the Robert Koch has done a lot of work in the area of science, diseases, uh, discovery of a few additional points in the microscopy. And there was one other important thing, I don't know which you mentioned or not, that he also observed the phenomena of acquired immunity, right? So he actually gave this concept, and he studied a lot of diseases such as plasmodium parasites, which causes malaria. Many other anthrax and many other diseases also. So a notable contribution to the science to the people by the Robert Koch, which is even today we remember that, even today we know that lot of basic research work in the area of bacteriology or the pathogen is being done by this great man. Yes. Okay, fine. So we have who is the next? Tanvi के बाद है त्रिदेव. नहीं है त्रिदेव. युक्ता.
Yes, ma'am. Good morning, ma'am.
Good morning,
Actually, I also studied the modification of postulates by Frederick and Bellman, which mentioned.
Okay, fine. Explain in your own words. It's fine. We can again hear that.
Yes. So they suggested the modifications of the postulate for the 20th century that the nucleic acid sequence belonging to present in most cases of infectious disease, and the nucleic acid should be found in those organs or the anatomical sites which are known to be diseased, not in those organs that lack the pathology. Fewer or no copies of the pathogen-associated nucleic acid sequence should occur in host or the tissue without disease. And with resolution of the disease, the copy number of the pathogen-associated nucleic acid sequences should decrease or become undetectable. With the clinical relapse, the opposite should occur. So when the sequence detection precedes disease, or when the sequence copy number correlates with the severity of disease, the sequence disease association is more likely to be a causal relationship. So next is the nature of the microbe inferred from the available sequence should be consistent with the known biological characteristics that proof of this can. Next is the sequence of which the such as the specific in situ hybridization of the microbial sequences in areas of tissue pathology and to visible microbes or to the area where the microbes are presumed to be located. So these sequence-based forms of the evidence for the microbial causation should be reproducible. That's it now.
Okay, Yukta, fine. Okay, so we can just stop this here and we can take up the next topic next week or maybe after that because we need to complete the syllabus also. So I'm not sure every week we'll be able to continue with the discussion part. And Adhish and Devanshi and Kshitij did not, and Tridev, Yuvraj. You'll get chance next time. And absentees me, I can see Vivek is absent. Then we have Athoiba also absent. Vivek and Athoiba are absent, right? Okay, fine. Just quickly give your attendance, right? Adhish Mishra.
Ma'am, sorry about earlier, I couldn't unmute myself.
Okay, fine. Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi.
Present, ma'am.
Ishita. Janvi.
Present, ma'am. Ma'am Ishita present.
जानवी का भी टर्न नहीं आया था शायद आई गेस शी डिड नॉट रिप्लाई राइट यस क्षितिज मानसवनी
Present, ma'am.
नम्रता
Present, ma'am.
नंदिनी नेहल
Present, ma'am.
नेहल
Present,
रूपल साहिबा
Present, ma'am.
संजना शक्ति
Present, ma'am.
Present, ma'am.
शिवानी तन्वी
Present, ma'am.
युक्ता युवराज त्रिदेव
Present, ma'am.
Present, ma'am.
Yeah. See, Kshitij and Athoba is not here. So, Kshitij, basically, you don't unmute yourself when I ask you a question or this. So this discussion is for you guys, right? So when you go and read something and when you discuss on the platform, there are 20 more people listening to you. This gives you confidence of speaking, of public speaking, basically. So this is more because I already know these things. So this is, or I might know new things from you guys if you've read something extra which I have not read, right? So what I believe is that this is more important for you guys. You guys are in 6th semester. You guys are next moving on to your master's degree or whatever job you want to do, right? So you should know how to present your thought. You should know how whatever you read and how you should be able to present that, right? So this is more important to you guys. So that's why I always poke you, I always encourage you to speak, come and speak, whatever it could be. It could be wrong, doesn't matter. We all are here, you all are my student, so it doesn't matter here. You need to be perfect when you're presenting, not in your class, when you're giving interview maybe, or when you're giving presentation outside for your job or something else. So there it will help you a lot, right? Okay, so I'll see you tomorrow, not tomorrow, maybe Monday for the next class. Thank you. Enjoy your weekend and stay
Thank you,
safe.
ma'am.
Okay.
Thank you, ma'am.